This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 326. Today is the 9th of January in the year 2025. Boy, the 2000s are moving along, aren't they? I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michelle Swanson.
Hello, good to see you all.
How's the weather there? Is it freezing?
Oh, it's seasonably cold. It's probably going to hit the low 20s, but we've got bright sun, so I'm not complaining.
Don't have a lot of snow?
No.
All right.
None.
From Charleston, South Carolina, Michael Schmidt.
Where it is also 4 degrees C or about 29.
Wow.
It's cold.
Yeah. Coming back today to be TWiM adjacent from...
Nice.
Hey, and that's very nice. Tacoma, Washington. Mark Martin. Hello,
it's
Mark.
a pleasure to be here where it's 21 degrees and 41% humidity.
21 Celsius?
Yes.
Yeah, very good.
So you're adding to our quality quorum today.
Yes. When Mark gets excited about a paper, we say, okay, come on and do it.
You bet.
If you're getting so excited.
It's fun.
And Happy New Year, everybody.
And
Same to you.
It's the first TWiM of 2025. Hopefully one of, how many weeks are there? It's 52, so maybe 25, 26 episodes.
That's what we've been averaging.
Yeah, it's good. I had to add them all up the other day for our tax filing.
Oh, my.
I had to count them manually because there's no program that will add them up. There's no program where I can say, how many episodes of TWiM did we publish in 2023? Nope, can't happen.
ChatGPT wasn't going to do it for you?
I don't know if I would trust
ChatGPT, yes.
That's the problem. They can give you this long answer, but how do you know it's right?
Sometimes it's true.
I'd like to go to a library and find a dusty old volume and find the answer in there. Then I know it's right. But people don't even like books anymore, right?
Well, I was very cross when our library got rid of all the books. It got rid of every book in the building.
I get the daily newspaper tossed onto my sidewalk each morning.
Mm.
Yeah, that's cool. I subscribe to the New Yorker and the Atlantic, so I can read it on the train.
Yeah.
And I'm the only person with a physical thing reading. Everybody else is reading on their phones, which is not bad. That's fine.
Oh, I don't know. It sucks the life out of you.
That's our rants for 2025.
Yes.
Well, you just wait till they put the wires in our head. That's what's going to happen next.
I'm going to run really fast, Mark. All right, we have science for you for sure. And remember, it's one word, plastics.
Plastics.
Remember that movie, The
Yes.
Graduate?
Yeah.
And Michelle's going to tell us all about plastics.
You bet. So the title is Landscape Profiling of PET Depolymerases using a Natural Sequence Cluster Framework. And this was published in Science on January 3rd of this month. And it's from a Korean team of Xiao, Hong, Park, Lee, Ki, Rao, and Sagong and Kim. And they are from Korea. And they were interested in the problem of PET plastics. So what are those? We probably are most familiar with them by that little triangle with the number 1 on various bottles that we bring into our home, beverages, etc. PET is a very durable material. It's resistant to a wide range of chemicals, acids, bases, solvents. So it's very good at for packaging material for food, beverages, pharmaceutics. It can also withstand temperatures up to 80 degrees C without losing its properties. The problem, of course, is that it does not decompose. It's been estimated that for it to naturally decompose could take up to 300 years. And I'm sure many of you have thought and worried about the amount of contamination. For example, in our oceans, it's estimated that there are 5 to 10 million metric tons have entered our oceans. And
wow.
This is a problem.
But also it makes particles, right? And you eat them in foods, right?
Yeah, that's
Little
the other
tiny
issue.
particles,
Nanoparticles.
nanoparticles.
Yep, microplastics.
Oh my gosh.
So colleagues, Yoshida and his colleagues from Keio University and Kyoto Institute of Technology in Korea went to a PET bottle recycling site and they collected 250 samples of sediment, soil, wastewater, and activated sludge. And published in 2016 the discovery of a bacterium that can completely degrade PET in about 6 weeks. They named this bacterium Ideonella sacchariensis. And they learned that it encodes two enzymes that can actually hydrolyze the PET plastic and generate then building blocks that that bacterium can use for growth. Others have found PET hydrolases in other organisms that they obtained from a range of environments. For example, the rhizosphere, which is a scientific term for the microbes that colonize plant roots. Also, mangrove soil, marine and wastewater sediments, and hot compost. So we know that there are bugs out there that can degrade these plastics. And this group decided, let's be really systematic and try to come up with some enzymes that we could actually use in industrial applications to intentionally break down accumulated PET plastics. So their first step was they turned to a database that is freely available on the World Wide Web that includes proteins that have alpha-beta hydrolase folds. So these include enzymes that degrade a number of complex molecules like polyesterases, lipases, cutenases, and it includes enzymes that have previously been found to degrade PET. So this was their starting point. So rather than go out and collect samples from the environment, they actually made a collection of DNA sequences, but specific to this particular class of enzymes, family of enzymes. So they identified about 2,000 sequences. Most of them were from two different families of bacteria, the Actinomyceto and Proteobacteria. And they first then took a bioinformatic approach, which they've outlined in Figure 1. And they applied a strategy called sequence clustering, which are basically algorithms that attempt to take this long list of different sequences and then group them by particular features that are related. So they're trying to deduce relationships. And they come up then with a network that we see in Figure 1. So it's kind of like a phylogenetic analysis. But of course, these aren't real organisms. They're DNA sequences.
Bravo. Thank you for saying that.
Yeah. And so they don't use terms like species and genus. We're going to find that they use other terms like clusters and nodes.
I should have said brava. Sorry.
Yeah. So they did this initial sequence clustering on these 2,000 sequences, and they got a fixed, not a huge number of clusters, and 3 of the clusters actually contained the PET hydrolases that had been described earlier. So this gave them confidence that the algorithms they were using were well tuned to their particular challenge. So once they had identified these candidates, they then took the next step, which was biochemical approach. So they selected 105 of what they call nodes, or candidates that were on this large network that's shown in Figure 1. And they expressed these 105 candidates in E. coli and then purified the enzyme and measured their capacity to degrade a powder form of PET, this plastic, at 30 degrees. And then they judged each of the 105 candidates by 3 criteria. How much monomer do they release from the PET? How much enzyme can be produced? And how stable is the enzyme to different temperatures? So on that primary screen, they found 5 of the best nodes or candidates, and they actually came from 3 different clusters. And so just the logic here is that bioinformatically, when they came up with clusters and found that they had some of the previously characterized enzymes, then it's reasonable to then take those as candidates, that they're likely to share a function. So that's the logic of this approach. They then did a second screen and looked for additional nodes in these 3 clusters and ended up with a group of candidates, 8 candidates that they wanted to test in more detail. So they looked and found that they had a number of candidates that could actually degrade this PET plastic at temperatures ranging from 50 to 70 degrees C, which is pretty warm. So again, their rationale here is they're thinking long term about this needs to be an industrial application. So that's why they're subjecting these candidates to these extreme temperatures. So two nodes that they want to follow up on are from two microbial strains. One is a micromonosporo species, and the other is from a Kutznaria species. And found in their sequence some depolymerases for this PET plastic. And these are called MIPA-P and Cuba-P. So they next got into more traditional or classical science. They first did some structure function analysis by determining the 3D crystal structure of these two enzymes. And then they took a genetic approach and did site-directed mutagenesis as a way to try to increase the thermal stability of the candidates they had. And they didn't just do this willy-nilly by just, substituting every amino acid with every other amino acid, because of course their number of candidates to study then just increases exponentially. Instead, they used a really smart strategy, which is they looked within their networks and their nodes and found particular motifs or residues that were conserved in the group. So they ended up with lots of different alternate amino acids that nature has basically tested because they're out there in natural species. So that restricted the number of site-directed mutants that they needed to make. They also added some disulfide bonds where it made sense based on the three-dimensional structure of these two enzymes. And that's because disulfide bonds basically act as struts that are known to increase the stability of the three-dimensional proteins. So with this logic, they ended up with 30 different variants of these two candidates of MIPA-P and Cubu-P, and they screened each of these 30 variants for stability at a range of temperatures, also a range of pHs, and they subjected them to, or basically measured their capacity to hydrolyze PET in a bioreactor. And again, the idea here is they've got an eye toward industrial application, so they're trying to move ever closer to kind of industrial conditions. So we see the results for those biochemical screens in Figure 5b, and then they screened for the capacity of each of these candidates to tolerate organic solvents. And again, this is typically important in industrial scale reactions, and so that's why they tested that capacity. And the winner in all this was a particular variant called QBPM12, and its features are summarized in Figure 6. But it has got the capacity, and again, these are engineered from the natural variant that they first found, but then they used smart predictions from what was known about other enzymes in this family to generate new candidates. So what they found is that this variant can tolerate at up to 70 degrees centigrade. It can tolerate the solvent ethylene glycol, and it can act on high concentrations of PET substrate in the reaction. And within 8 hours, it can depolymerize 90% of the PET plastic. And importantly, the enzyme was able to maintain activity for as long as 10 days, even at temperatures of 40 to 50 degrees. So they conclude with this and are optimistic that this is a great strategy and have demonstrated with this kind of proof of principle by engineering this QBPM12 variant, that this is a realistic approach to identify and generate new enzymes that could be used in industrial applications to degrade PET plastic down to its constituents, which could then be reused, for example, or stored some other way.
So, Michelle, currently when you recycle this stuff, what happens to it?
It's made into planks.
Planks?
Planks for picnic benches and things like that.
Yeah.
Yeah, it's also being used to make different fibers. So some clothing can be made from it. But the amount that we're generating and using in our, soda bottles and that is nowhere near the amount that's going into this recyclables.
So the use case here would be to throw a bunch of bottles in a big vat and to put this enzyme in and have it digest all the bottles, right?
Yeah.
Basically.
I think that would be a great start, because ideally we move away from these plastics that don't biodegrade and move more toward glass or metals. But yeah, we do have this problem of tons of PET in our oceans, for example.
So this candidate, Kubu PM12, is this final, or do they still need to do some tweaking?
I would imagine that they have some other things they want to optimize, but I honestly don't know enough about the whole process of doing this breakdown in bioreactors to answer your question.
Yeah, okay.
Their approach reminds me when I was in graduate school in the last century. This reminds me of how they were selecting enzymes that were incorporated into laundry soaps. And they were using many of the enzymes that bacteria have, and they were going after the thermophilic microbes because their hypothesis was that thermophiles would have enzymes that would be more stable. And if you think about it, 70 degrees is much warmer than routine laundry that we conduct here in the United States. Not so much in Germany. Germany, they effectively use very, very hot water to launder their clothes. But here in the States, they've been adapting the enzymes to work at cooler and cooler temperatures.
Yeah, cold temperature I try to use.
So this is a really fascinating approach. And what I really liked is they're being able to systematize this to effectively identify the cluster that would give them the biggest bang for their buck. And in one paper, they were able to do a lot of science that really holds promise. I'll bet you they're trying to figure out how to immobilize this enzyme on a surface so that it retains its activity, so they can flush out the tank, get the monomer, and do this in a continuous rather than a batch-based process and lose their enzyme.
I share your optimism, and I do want to give a shout out to Korea's Ministry of Trade, Industry, and Energy and their National Research Foundation that funded this research.
I think it's interesting that they say in the introduction that these bacteria have enzymes that are pretty nonspecific and can digest. Because if you're wondering, how does this digest a human-made plastic, right, which doesn't exist? It's just because the enzymes are pretty nonspecific. Then you can improve them as they have done here. Yeah. So that's pretty neat.
For many years when I was teaching micro, I would talk about the principle of microbial infallibility. So if nature made a particular type of a molecule, microbes could break it down, sometimes not by themselves, but they could do it. And one exception at the time were lignans. And the issue there is relevant because the subunits of lignin are assembled not completely randomly, but almost randomly. So it's really hard to come up with an enzyme that could attach to those and then cleave them. So in fact, some of the white rot fungi just make a whole bunch of peroxide and just like tear it apart. But this is different because they're actually looking for things to kind of fine tune that active site, as Vincent was saying, and that makes it remarkable.
It's very cool.
I'll also add that I presented as a snippet, so I kind of kept it at a high level, but this was a lot of work. Imagining more than 100 candidates, they make an E. coli, do the quantitative assays, thermostability, et cetera. This was a heroic effort, and I applaud the authors.
More evidence of the awesomeness of microbes.
Yeah, they started with microbes.
Come on, first evolved, last extinct.
Yeah, that's right. Thank you, Michelle.
My pleasure.
Mark, what do you have for us?
Oh, man, I have some wonderful stuff for you. I'm so excited. Let's see if I can do it in a way that my colleagues really appreciate. This is all about stress responses by bacteria to the environment. And I'm reminded of when I took ecology, this idea of alarmones, some of the ideas of what's signaling. So the title of the paper is Bacteria Use Exogenous Peptidoglycan as a Danger Signal to Trigger Biofilm Formation. It was published on the 3rd of January of this year, though it looks as if it sat in reviewers' heck for a while. It's
October 22. Oh, my gosh.
Wow. Yeah.
They made him go back to the bench is what I think.
Well, I'm quite sure. And there's a saying, about beating a dead horse. What I think they had to do is go back and really hammer things down. So I will tell you in advance that this is a paper I am going to be having my micro students in the fall go over, and I'll explain why when we come to it. I will say very small print. So this is a paper that really deserves to be on a big screen. First author is Sanika Vaidya, and then there's Saha, Rode, Torrens, Jekyll, Gittig, lots of different people, and they're mostly European. As I look it over, they're from Marburg, Germany, Basel, Switzerland, Umeå, Sweden, Frankfurt. I'm almost saying that correctly. Even some Copenhagen in there. And
Hagen. Elio says it's Hagen. You don't make German word out of it. That's what he did to me. He said, Vincent, it's not a German word. It's
you
Hagen.
would have definitely gotten yelled at.
I was. He yelled at me for saying Copenhagen.
He did. I remember. I was there.
So sorry, Mark. Copenhagen.
Elio would not have yelled at me. He was always kind to me.
Well, he didn't yell. He just said, Vincent, it's not a German word.
Yes.
I miss him. So, and there is one person at Northeastern, which is interesting. That's the one American out of there. So,
so it's Umio.
Okay.
That's another. I mean, I get told by people how to pronounce these things. So.
And so this is a paper in Nature Microbiology, a journal I quite like. And no, I don't receive a free subscription, and I'm angry about it. But let's go ahead and go through it because it's a lot of fun. This is all about danger sensing. And the first thing that I want to remind everybody is we have to talk about biofilms. And it's a very interesting approach to it because most people who are microbiologists are pretty sure they know why biofilms exist. And biofilms are so ubiquitous, they're so involved in so many things, they can't have just one purpose. But let's revisit that at the end of this paper. So this is all about sensing danger, and it's about how some interesting results came up with a group who was studying how bacteriophage are interacting with Vibrio cholera in a microfluidics device. The hope was maybe bacteriophages could be used to impact the transmissibility of this pathogen, but instead, these researchers found what they called a danger signal. Now, just to give it away a little bit here, it turns out that peptidoglycan from a variety of bacteria was this danger signal, and the response was increased biofilm production, ostensibly protection against bacteriophages. So the first thing I have to do is remind listeners that we're talking about peptidoglycan, which is part of the cell wall of many, many types of bacteria. And what I tell my students to think about it, peptidoglycan, these are polydisaccharides made of N-acetylmuramic acid and N-acetylglucosamine, and then they have short peptide chains linking those together. And those short peptide chains are fascinating to poor old Doc Martin because they contain D-alanine. So the whole idea, and what I tell students to remember about peptidoglycan, think of them like bacterial Spanx. And what they deal well with here is the idea of osmotic changes. So they help, it's like a network or a knit bag surrounding the cell membrane. So
nylon stocking.
Yeah. Spanx may not be a widely understood term.
Well, I'm when I say the nylon, the fishnet stockings, where this goes to student cartoons that I can't reproduce. Okay, but I like that too. But it's actually
Martin
quite true.
Wearing fishnet stockings.
No. I have drawings by students of bacteria, kind of dressed like they're in cabaret.
Okay.
Maybe compression socks.
Yes.
Nice. I like that. And and this is a very exhaustive paper. There's a lot in here. And it turns out that, like I said before, this whole issue has to do with peptidoglycan, and I'm going to circle back on that toward the end. So I know that most people, and I was one of them, used to think about biofilms primarily as a way of staying in one place so nutrients came by. I see this all the time. Or protection, as some people think of them that way. And so that's what I was thinking as I got into this. And what I really like here, and I'm going to refer to some of the figures here. They were using a microfluidic chamber, and what they did was allow Vibrio cholera to form some adherent cells. And then they added phage to wash it through. I will say they call LB Luria Britannia broth, and we know that's not what it's called, but I've said it many times too.
What is it? What is LB?
Lysogeny broth.
So why are they allowed to call it Luria Britannia in a peer-reviewed publication?
Because the editor and the reviewers didn't correct them.
They didn't know, yeah. Lysogeny broth, yeah.
A lot of people call it Luria Britannia broth.
I am sure that's the only weakness in this beautiful paper.
It is beautiful. Thank you for saying that, because it is in what and as we talk about the amount of time it took to get this out there, you can just see that they took like a tennis shoe and beat out every possible fire that anyone could come up with.
And if I could interject, we've been in touch with the first author, Sanika Valadi, and she mentioned that she saw the initial result in the very first month of the project.
Yeah.
But it was a long journey to figure out what was happening.
And this is something I want listeners to really appreciate. When you get a completely cool result, you have to science the heck out of it.
Oh, yeah.
And this is where peer review is so important. Even when you don't like reviewer number 2, it's very important that we understand that this is how science progresses. So in any event, what they're allowing, and I've seen this done in many biofilm papers, is they allow a biofilm to begin to form in a microfluidic chamber, and then they use a wash. And it could be enzymes or whatever. In this particular case, they were using bacteriophage. And this is a specific bacteriophage against cholera, Vibrio cholera. And most of the cells were lysed quickly, but the survivors rebounded and formed 3-dimensional biofilm foci. They call them colonies. And I don't know if that could really be the proper word here, but they're certainly foci of growth.
Microcolonies? And it was microcolonies,
I
maybe?
guess. Yeah. But they're really interesting because they used, to get ahead of myself, they used wonderful imagery to actually visualize these things.
And to show them to us.
Yeah. Pardon me?
And show them to the reader.
Yeah.
Yes.
Lovely figures.
And this is like figure 1A will do this. Now, what's really interesting here, and this is where observation is critical. You know, biofilms take lots of different forms. And what they do here, which is so much fun and remarkable, is they actually draw a distinction between a 2-dimensional biofilm and a 3-dimensional biofilm. And this is important. So how do they do that? They had to measure things that were above 3 microns. A biofilm that's above 3 microns very clearly is a 3-dimensional structure. Things under that they defined as a 2-dimensional structure. So to repeat myself, when they added the bacteriophage, and this is at a multiplicity of infection of 1, most of the cells, again, were lysed quickly, but the survivors rebounded and formed these 3D biofilm foci. If you do this in the absence of phage, you just get those 2-dimensional biofilms. And this is, again, figure 1 has so many wonderful pictures. And if you look at those resulting organisms, you might suggest that they might be mutants to some form of resistance or better able to hold on to the chamber. But in fact, that's not true. They were not phage resistant, nor were there biofilm hyperproducers. And they even went to the trouble of confirming that quorum sensing was not involved. Now, they did these things without really making mutants to do that. They just would look to show whether or not the survivors were resistant to phage. And they used what's called a Rogos phenotype to show biofilm hyperproducers, because that's a well-known system in Vibrio. But the interesting part is in figure 2, a phage-free cell lysate. And that means this is not a lysate that was made from bacteriophages as they lytically infect and burst cells, seem to also cause biofilms.
So just a mechanical disruption,
yes.
Sonication to get a gamish. Then they add the gamish.
I'm so delighted to hear that word. It brings me back to my late mother, gamish.
And they saw the same remarkable phenotype.
Yeah.
And they could have probably done it with penicillin, too.
Well, we're going there, Michael. were going there, surprisingly. Now, they also said, okay, what if I use heat-inactivated phage? Well, no, they didn't get that effect. They also worked with mutants that were involved with cell lysis, and that also didn't seem to work. Again, the sonicated cell lysate seemed to lead to biofilm formation acting the way phage did. And it was a 10-minute exposure is all it took. This is pretty remarkable stuff. Now, again, figure 2 is worth your time. As I look at it here, as I'm speaking to you now, oh my God, figure 1 is beautiful. I mean, figure 2, if you look at it, I mean, look at 2A, for example. They have phage exposed and look at those differences between unexposed, active phage, and inactive phage, what the effects are.
Yeah, you don't need any stinking statistics for that.
It's beautiful.
But they gave us some. They gave us stats.
They have to. You have no choice.
Yeah, I believe that might have been some of the extra time that was required for this. So what's interesting here to me is that they were trying to figure out what about the gamish was leading to this effect. Again, just a 10-minute exposure, and you have something that's easily definable as a different biofilm phenotype.
So interesting. They're imprinting on those survivor cells to switch and their developmental program to biofilm formation after just 10 minutes.
It does seem to imprint itself as, and that's a good term for it, for several generations afterwards too. It's really quite remarkable. So what they found when they went through this, it was I can't talk today, I'm so excited, peptidoglycan, which I call PDG, that seems to be doing this. So it was the peptidoglycan in the cell lysates that made that happen. So they thought, does it have to be from Vibrio cholera? And it doesn't. They checked a number of gram-positive and gram-negative cells, and the cell-free lysates of all of them, if you go to figure 3, again, a thing of beauty. Would you just look at that?
It is beautiful. So Vibrio cholera, E. coli, Pseudomonas, Bacillus subtilis, Staph aureus, all of their peptidoglycans have the same effect after just 10 minutes.
I like the use of spheroplasts to prove that.
We're almost there. We're almost there. Okay. Now, I have to be fair about this. Now, once again, lysates of a variety of these bacteria all led to this. So they wanted to know, okay, what are we going to do with something that doesn't have peptidoglycan? And that would be a good thing to do. Not perfect, but if you make spheroplasts, by basically using penicillin to prevent that peptidoglycan from forming and keeping them under osmotic stability, you can create a population of cells, and they look like blebs because they can't hold their structure very well, and that's why the sugar solution keeps them in play. And they don't get as big of an effect from that. And I believe, that that's because peptidoglycan is still attempting to be being formed under those conditions. They didn't check L forms, and these are the bacteria that can occur that don't appear to be making peptidoglycan at all, but they didn't have access to that. They did, however, do exactly what I taught my students to do with this kind of stuff. They tried to digest peptidoglycan with enzymes, and they found that it removed the biofilm producing impact. And so in order to do this, they had to use purified peptidoglycan. It turns out you can buy it, but it's made from Bacillus subtilis. But that's no problem because it still has this effect. Questions or comments so far on that?
That must have just been such an exciting time in the lab. You know, to go through this systematic work and then to purchase some peptidoglycan and boom, same phenotype.
I agree 100% with that.
So let me get this so far, Mark.
Sure.
Along come phages, and with them is peptidoglycan, and the bacteria that are about to be killed by the phages. And nope, we sense peptidoglycan. They make a biofilm to increase resistance. Is that the idea?
That's the idea.
And that's because the Vibrio phage has autolysin that causes the bacterium to lyse. And you don't get this enzymatic digestion of peptidoglycan. You just get the explosion of the cell because it's autolysing. It literally releases the progeny phage into the surrounding medium.
Right. So that the phage aren't killing the bacteria just to be ornery. It's once the phage have multiplied, then they need to get out and go find a new host. So that's when they autolyse them.
And it's a really simple enzyme system to say, okay, we're done, let's explode. And that's what goes on.
I get criticized all the time by students for inferring intentionality to microbes, but there we go.
But
right?
we're old, so we are allowed to do that.
Well, it's fitness.
It's fitness.
Yeah,
Well,
it's fitness. Same thing with viruses.
I
It's all
just
about
say
fitness.
that first evolved, last extinct. That's my comeback to that. So this brings us to figure 4, which is really exciting to me. They knew that it looked as if peptidoglycan was increasing this molecule that is freaking everywhere in microbiology. Cyclic di-GMP is a kind of a global response ligand, and also often is associated with increased biofilm matrix production. So if you go to figure 4, they did RNA-seq. They did it with and without peptidoglycan. And they found that 325 genes in Vibrio cholera were upregulated and 70 downregulated.
In response to peptidoglycan.
And what was so cool about this is that the Vibrio polysaccharide VPS itself is necessary for those 3D biofilms, and the genes involved with that have been upregulated, as were phage defense genes, as was RPOS, and the diguanylate cyclases. What excited me no end, and I apologize for my enthusiasm, is they had a fairly complex description of a reporter gene that basically could indicate in living cells the levels of cyclic di-GMP in the biofilms.
So they could watch it under the microscope in real time.
That's right. And I'm so excited by this because mostly we have nucleic acids that we look at and we make guesses about it, but they can see it. And this is only because of all the hard work of many, many researchers developing Vibrio cholera as a model system and the knowledge of many other people that was brought to bear on this.
And
however,
go
ahead.
Folks should know that what RPOS is a secondary sigma factor. So as you appreciate that sigma, it controls what genes are transcribed at any given moment in time. By the virtue of the fact that it's upregulating RPOS, that's effectively telling, or if you will, as using Michelle's phraseology, it's imprinting the activity for future.
I agree completely. Now, one thing that they really made a big deal about, and I was so delighted to see it is not clear how Vibrio cholera senses peptidoglycan. I would tell you that it must be able to sense it as part of biosynthetically creating their own, right? That clearly has to exist, but what it is, how it's being quote seen, perceived, is not yet clear. Now, most of this has been me talking excitedly about cool things that they did. And I want to add one more, and it's figure 5. And I really want to emphasize this because many people talk about what biofilms do. They very seldom measure it. In figure 5, the authors were able to show that these peptidoglycan-induced biofilms appear to protect against bacteriophages. And the way that they did that was not the way that I would have. They're so much smarter. They used a phage infection reporter. So they're not measuring new phage. They're showing of the genes that are turned on in the cell during phage infection are being activated by this. So in other words, if you have been exposed to peptidoglycan, those reporter genes are turned down. And this demonstrates that they are protecting against bacteriophage. It is
so it's a nice kind of closing the loop. They started out with this observation that the phage trigger biofilm formation, and now they take it to the functional test. Okay, if you form a biofilm, does that confer any advantage? Yeah, it makes you resistant to phage infection.
Now, okay, so what's interesting here is figure 6, and it's amazing. It doesn't work perfectly for everything, but what you see is that PDG is a danger signal. There needs to be a more elegant term. I can't think of it right now. This alarmone. I remember when someone did an experiment many years ago when Dello Vibrio infected E. coli and they measured the upregulated proteins. They called it a bacterial scream. So we need something similar here. So if you look at figure 6, they show that PDG is a danger signal across different species, leading to increased biofilm production. You certainly can say, yeah, it's Vibrio cholera. That's what we're working on. Look at Pseudomonas aeruginosa. Look at Acinetobacter. Look at E. coli. Look at Staphylococcus aureus, right?
Yep.
Now, Staphylococcus epidermidis could be better, but at least they included something that didn't, follow as dramatic a difference.
Yeah, they showed all their data.
They
I
showed
admire
all their
that.
data.
Mm-hmm.
It is, I want to say that if you read the paper, there is a lot of technical detail. They really were exhaustive about it. And I would use that word. It must have been exhausting to do this, but also.
And so well controlled, really systematic, very well controlled, showing all their data, their logic. It's amazing.
Their materials and methods are peerless. Their additional information is worth your time. I do want to just kind of close out before I get discussion with you all about this with two things, and it's this. Now, peptidoglycan has long been known to act as, in the olden days, we called them PAMPs for pathogen-associated microbial patterns. And because these also occur in mutualist associations, many people, like Margaret McFall-Nye, suggest we call them MAMPs for microbially associated molecular patterns, certainly recognized by the innate immune cells and having responses. So we know how those work. But there's a final thing, and I actually put a link to this paper in the show notes. Bordetella pertussis causes pertussis, whooping cough, and it has a really terrible effect. And it turns out that there was something in the culture supernatants of Bordetella pertussis that led to this, and it was called TCT, for tracheal cytotoxin. And nobody knew what that was. It had that horrible effect on the throat. And I hope pertussis doesn't come back. It's not a good disease at all.
Yeah, it is right now.
It's back. Big time.
Right. A lot of cases in the U.S.
However, it turns out that if you look at the Hawaiian bobtail fish, Hawaiian bobtail squid, I corrected myself, Michael. Relax.
Well, I'm just doing Margaret. I'm just doing Margaret.
So these people are really mean to you folks. They're pretty nice to me. I like it that way, by the way, Michael. So in any event, Euprymna scalopes, the Hawaiian bobtail squid, makes a light organ, and it only does it when it's around Vibrio fischeri. Turns out that if you put TCT from Bordetella pertussis in with the squid, they develop light organs. So this is suggesting that fragments of peptidoglycan are signal molecules in all kinds of complicated ways. I would also add, the authors did not mention this, but I know from a lot of work that's been done in unusual Vibrios that many Vibrios tend to shed fragments of peptidoglycan. I don't know that every bacterium does that. I happen to know that Vibrios do. And so the idea of them constantly producing this would work against the idea of this danger file, although there's evidence in the paper of a dose-dependent relationship. So there may be kind of transition concentration that gives rise to the formation of the actual three-dimensional biofilms. But again, I want people to really remember that the microbial world is full of signals. I don't know if it's original with Joshua Lederberg. That's kind of interesting to think about. But he used to say that microbes are talking all the time. We just don't know the frequency. And he said that long before quorum sensing. But I think it's still relevant that we tend to forget that they're always signaling. I myself wonder about the amount of peptidoglycan that's released naturally during cell division. That has to be below the threshold that causes this to happen.
I think you got to also look at the death phase, because this is a population effect. It's not an individual cell effect. And I think oftentimes microbiologists suffer from thinking about a single cell because we think of bacteria as individuals when they are in reality a community. And every first year micro student can effectively draw a bacterial growth curve. And the hardest thing for them to appreciate is that it's a population and not an individual.
Well, and biofilms are a great example of that because there's division of labor within the biofilm. So different cohorts of cells are doing different jobs.
And I went down the rabbit hole of thinking about figure 6, specifically why Staph aureus was so high with its signal. And I wondered if that was because Staph aureus cross-linking is actually pentaglycine. It doesn't have your D-alanine. And so, but, whether or not that was just a fluke of the way the figure was presented or whether or not it was a big deal because it was pentaglycine, because the only thing different between the peptidoglycans is the cross-linking. Because, it's N-acetylmuramic acid and glucosamine, and that's peptidoglycan. What's different in peptidoglycan is the cross-linking amino acids. And in the case of Staph aureus, it's pentaglycine. And remember, another staphylococcus has an enzyme that breaks down pentaglycine, and that's lysostaphin, which was actually used as a therapeutic.
So this is interesting too. And I'm glad that you're mentioning that, and also that Michelle is talking about the fact that when we study single species biofilms, we have to be very cautious. At the same time, that has to be how you begin the work.
Sure.
Right? Oh, and one thing that just occurred to me, do you all remember this? Oh my goodness. A lot of bacillus, and I can find this link and send it to you if you want. A lot of bacillus, if they're being preyed upon by Myxococcus, make these castles. Do you remember this?
Mm-hmm.
And they're doing this in response to predation. And of course, when I'm thinking about this, because Myxococcus with the wolf pack is just breaking down everything. So maybe that's related to this again, what these danger signals are.
So one implication that I'm concerned about, this is really beautiful biology, microbiology, but there's some implications for bioremediation. So the bacterium that your tax dollars have funded me to study is Legionella pneumophila, which causes pneumonia, but we don't share it with each other. We get it from contaminated water. And what's known is that if there are buildings that are colonized by Legionella and they've had cases associated with it, then the team comes in and they do a heavy remediation to clear the Legionella. But often what they find out is months later, the same lineage regrows. So there was a case of a long-term care facility in Illinois, I think it was, where after years and years and years of trying to get rid of the Legionella, they ultimately decided they had to just knock down the facility and build a new one. But what it suggests to me is if the biocides that are used, to get rid of Legionella in water systems lyse the bacteria, and if Legionella has a robust response like all these other bacteria do, they're basically going to just buckle down, form biofilms. They're now resistant to the biocides, and that could explain why it's just not possible.
Why it becomes persistent.
Yeah. So how do we kill Legionella without it releasing any peptidoglycan to trigger this perhaps conserved pathway?
Or after you use the bleach, you go back and you use another disinfectant. Perhaps you use radiation or high ozone concentrations in the water in order to see if you can get rid of any of the persisters, where you're actually going after to fracture the nucleic acid into kingdom come, the molecular grenades, if you will.
Yeah, which of course is harder to do if in the first 10 minutes they form biofilms.
Yes.
Once a biofilm is there, it's a lot harder to clear.
Michelle, I'm so glad you adored this paper the way I did. But did you notice, how you could take almost any figure and spend a whole class period with that?
Oh, for sure. Yeah, we did learn from the first author. And so the first author is named Sonika Valdia, And she tells us that her first lab experience was in a lab cultivating soil microbes in high school in India. She was so fascinated by what she saw under the microscope and also loved kind of the logical reasoning about forming hypotheses that she committed at that time. I want to be a scientist. So she got her bachelor's in microbiology in India and got a scholarship then to do a master's degree at the Imperial College of London. And there, she wanted to do interdisciplinary work for her PhD. So she joined Knut Drescher's lab at Max Planck Institute for terrestrial microbiology in Marburg, Germany. And there she learned to think about biology from a physicist's perspective, which I'm sure came in handy thinking about these biofilms. She then wanted to return to her roots and work toward developing new drugs. So she has now moved to Kim Lewis's lab at Northeastern University in Boston and is currently cultivating soil microbes now as a starting point to find new biotics. And she says that each step of her education and research, she's had great mentors, and she's still really grateful and still looks to them for career advice. She does remember that in this project, which lasted more than 4 years, everybody contributed. She had great scientific discussions with lab members, including the postdoc who mentored her, Praveen Singh, and also helped her shape ideas and prioritize experiments. She also collaborated with Eric Jelly, who was a fellow PhD student, an amazing physicist and programmer. She says the most exciting day was when they figured out that there was peptidoglycan fragments induced biofilm formation. She excitedly called her parents that evening to tell them what they found. And of course, they didn't immediately understand it, but they were enthusiastic, as most parents would be. She does have advice for aspiring scientists. Trust your gut bacteria and follow your instincts. Find that part of science that excites you, that thrill you get from a successful experiment or from your hypothesis turning out right. It's unlike any other kind of material monetary compensation. While doing research, also do something else. Pursue a hobby, a sport, something that stimulates a different part of your brain. And for her, she likes to play board games, learn a new language, go ice skating. And then finally, she says, surround yourself with people who exude positivity. Congratulations on this lovely work.
It's wonderful.
Thank you, Michelle. Thank you, Mark. Really cool. All right, that is TWiM 326. You can find the show notes at microbe.tv/TWiM. You can send us questions or comments, twim@microbe.tv. And if you enjoy these programs where we discuss science, real science, not fake science, we'd love your financial support, microbe.tv/contribute. Our TWiM adjacent, Mark Martin, the University of Puget Sound in Tacoma, Washington. Thank you, Mark.
You're very welcome.
Michelle Swanson's at the University of Michigan. Thank you, Michelle.
Thank you all. Great to see you again.
And Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.